Good morning, students. Let us start the revision of ribosomes. Now, ribosomes is a cell organelle present in both eukaryotic as well as prokaryotic cell. And I have told you that there are two types of ribosomes, 70S as well as 80S. Now, each ribosome has two subunits. One is the large subunit and the other one is the small subunit. Now, normally, in normal condition, the two subunits remain separated. Now, when the ribosome perform the process of protein synthesis, these two subunits combine together and attain its structure. Now, children, how this process occurs or what does the ribosome do? Actually, the ribosome is the site of protein synthesis, as you all know, and proteins are made up of amino acids. There are approximately 21 type of amino acids present in the living organism. 21 types. Now, out of 21 types, 9 amino acids cannot be formed by human body. So, these 9 amino acids are taken up from plants. Now, children, these amino acids combine together. It can be like, suppose this A1 is an amino acid, it can combine with A3 amino acid, it can combine with A5, again with A1, or any order and attain a particular structure and form a particular protein. Now, after the joining of these amino acids, a chain is formed. That chain is called as polypeptide chain. Now, children, this polypeptide chain, suppose this is a polypeptide chain. Now, this polypeptide chain make a particular structure and that structure is called as protein. Now, how this polypeptide chain attain a particular structure? Now, this chain wound around any cofactor. Cofactor can be any inorganic compound or it can like metal ion. Suppose I said zinc. So suppose this is a zinc and this polypeptide chain is wounding around itself and attaining a particular structure. Now this is a protein. Now there will be bonds between the amino acids present in the chain also. Now suppose this polypeptide chain is wounding around coenzyme. Now coenzyme are the organic molecules. Cofactors are the inorganic ones and coenzymes are the organic molecules like vitamins which catalyze the reaction. So this protein, this polypeptide chain will wound around itself and attain a particular structure. Now children, if this structure is disrupted, अगर हम इस स्ट्रक्चर को डिस्टर्ब कर दें, इसको खोल दें, तो ये अपना फंक्शन परफॉर्म नहीं कर पाएगा. दिस मीन्स that for a protein to perform its function, the structure is very important. If the structure is broken, it will not be able to perform its function. I think this is clear to you now. I can give you an example of a gram seed. चना आप खाते होंगे. तो when you are going to soak that gram seed overnight in water and after removing the water next day, this gram seed can germinate. Now if you boil that seeds, if you have heated that seeds, then it will not be able to germinate. Why? Because the enzymes present in the gram seed has lost its original structure. उसका जो original structure है वो disrupt हो चुका है, जिसकी वजह से अब वो क्या कर रहा है, अब वो अपना function perform नहीं कर पा रहा, जिसकी वजह से germination possible नहीं है. This was about the proteins. Now children, you have studied about central dogma of molecular biology in the video related to the introducing biology. Central dogma. In central dogma, I have told you that DNA forms RNA and from RNA proteins are formed. Now the formation of RNA from DNA is called as transcription, and the formation of proteins from RNA is called as translation. So children, what happens here in ribosome is that when these both subunits join together and starts the function of the formation of protein, RNA comes and binds here, and this is responsible for bringing the amino acids which bind to its site, and then they are responsible for forming the bonds between the amino acids. Now children, I don't want to go in much detail, but still I just want to mention here that RNA are of three types. That is ribosomal RNA. It is present in ribosomes. tRNA is transfer RNA. It is responsible for bringing the amino acids to this site, to the ribosomes. And then mRNA. This mRNA is responsible for telling that which amino acid has to be brought to the ribosomes. Children, again listen to me very carefully. I am telling you that RNA are of three types. mRNA, that is messenger RNA. Now this messenger RNA is responsible for instructing or for telling that what amino acid or which amino acid has to come here and be a part of the protein. Now again the second one is the ribosomal RNA. Now children, this ribosomal RNA is the constituent of the ribosomes. The last one, that is tRNA, is the ribosome, that is the transfer RNA, is the RNA responsible for bringing the amino acid to ribosome as instructed by mRNA, as told by mRNA. एक बार फिर से सुनिए, मैंने कहा कि RNA तीन टाइप के होते हैं. mRNA जिसे हम कहते हैं मैसेंजर RNA, tRNA जिसे हम कहते हैं ट्रांसफर RNA, और rRNA जिसे हम कहते हैं राइबोसोमल RNA. mRNA का काम होता है कि किस अमीनो एसिड को लाना है ये बताएं. इसके ऊपर कोड्स प्रेजेंट होते हैं, इसी पर राइबोसोम बाइंड करता है. tRNA का काम होता है कि जो mRNA ने राइबोसोम जो mRNA अमीनो एसिड बताए हैं उसको लेकर के आना साइटोप्लाज्म से सेल के. और राइबोसोमल RNA जो होता है वो राइबोसोम में नॉर्मली ऐसे ही प्रेजेंट होता है, उसका एक कॉन्स्टिट्यूएंट होता है. इज इट क्लियर नाउ? अब बच्चों प्लीज फोकस हियर दैट दिस इज नॉट इन योर सिलेबस, बट योर कॉन्सेप्ट विल बी क्लियर बाय नोइंग दिस. No need to write these things in your exams like in class 9th or in class 10th. Is it clear? Now let us move on to the next slide now. You can see here about the ribosomes, eukaryotic as well as prokaryotic. The eukaryotic ribosome is 80S, which is made up of two subunits, 40S and 60S. The prokaryotic ribosome is 70S, which is also made up of two subunits, 30S and 50S. Now 50S is the largest subunit in 70S and 60S is the largest subunit in 80S. Now I think this S is clear to you all. In the previous video I have explained you about that S is responsible for SWID work. That is the SI unit of sedimentation coefficient. Now moving on to the next slide. Ribosomes are the site of protein synthesis. Ribosomes are numerous small granules either scattered freely in the cytoplasm or attached to the membranes of the endoplasmic reticulum. These are the factories for the synthesis of proteins. I have told you how. So children, that this was all about the revision of ribosomes. If anything is not clear to you, you can mail me at mithalisharad7@gmail.com. I have mentioned my email ID in the description box. Please don't forget to like, share and subscribe. Thank you. That's all.